this half an hour on talk on shelf life enhancement technologies in millets. My overall presentation will be on bottlenecks in shelf life of millets, post-harvest losses in millets, and how to manage them, and finally the technologies for enhancing the shelf life of millets. I'll have a brief introduction. I think all these days you have a lot of introduction on millets and all that. I'll just skip this slide, but just to know that global millet production is estimated at 28.37 million tons, and over 50% of the whatever the millets are produced is presently now finding its use as alternate resource than its consumption as a staple food. Um, so if you see, I think many people would have talked about the nutritional and health benefits and what how important the millets are. Yes, we agree that they are an excellent source of carbohydrates, proteins, dietary fiber, vitamins. So many minerals are there, and despite of all these numerous advantages, the major challenge is the processing and the storage of the millet flour. That is called the shelf life of the flour and the grain. So if you see here, I have just made what are the bottlenecks in different millets regarding the grain. You have dehulled grain, flour, and the products, value-added products made out of these millets. If you see the pearl millet, the major problem is with the grain, flour, and the products. Foxtail millets, it is problem, the shelf life of a dehulled grain. Once, if it is a grain, yes, it is stored for many, I think, years. But once it is dehulled, the problem, the shelf life is a problem, and even in the products also. In finger millet, flour shelf life is a problem. Barnyard millet and kodo millet, again, we have a problem in once it is dehulled. And sorghum, we have problem in grain, flour, as well as the products. So, what is a scientific studies on storage of sorghum and other small millets have been very less compared to the other cereals. Why? Because an important reasons might be because of the traditional practices what the farmers are following to store the millets. Usually, they store the millets in structures like I'll just show you, like bins, storage method like mud structures and all this. These are cheap and constructed with locally available eco-friendly materials that considerably sometimes increase the shelf life of grains. I'm talking about the grains, not the flour. But you can see the shelf life of the millets is usually affected because of insects, microbes, as well as microbial biochemical degradation. I have given some examples here. The first one is if you see the pearl millet, next the foxtail millets, even the sorghum, it is damaged by the insects. We'll come into the part later. That was the grain shelf life. Now we're coming to the flour as well as other products like ragi flakes. You can see it's spoiled very fast here. So even the signs of symptoms will start appearing in 10 to 15 days of storage, even at the ambient conditions. But usually the traditional methods what farmers is practice is they reduce the insect or pest attack by locally placing the neem leaves or dried red chilies inside the bags. Mainly the neem leaves act as an antifeedants. So they give protection definitely if you put the neem leaves layers and layers. In this way, the keeping period of millets can be extended. However, these grains, as a grains, you can protect them from the insects by properly storing them. But once it is dehulled or milled, you call it, then because then the shelf life is reduced, because milling what happens is it breaks the cell wall and destroy the seed structure, what you say. So which will attack, because of the destruction of the seed structure, the seed is attacked by the enzymes, microbes, insects, so many other environmental factors. In India, mostly the post-harvest losses are caused by unscientific storage, because of insects, rodents, microorganisms. These all together account to about 10% of the total food grains. If you see this, the insects post-harvest losses, 12% are before the harvest and 37% after the harvest. That is, post-harvest losses are more than the field losses. So together, the 51% is the major economic losses caused by the insects. Because the actual material they consume is more than the damage they impart. The amount contamination and the excreta, these are all make unfit for human consumption. About, if you see, there are about 500 species of insects, which we need not bother about all the insects. Nearly 100 species of insects are causing damage to the stored pest, but very few are of economic damage. So if you see the post-harvest losses in India, they are account to about 12 to 16 metric tons of the food grains. And in the monetary losses, if you see, it is about rupees 50,000 crores per year. So the majority of the farmers estimated that sorghum yield losses go up to 50% due to insect damage during storage. So mainly because of all this is a lack of hygiene storage conditions. If you see, just I will give a small brief introduction on the insect pests. You can see I have put the figures. These insects not only attack the millets but also other cereals like rice, wheat, and all that. These are divided into two types, like primary or internal feeders and secondary or external feeders. Primary internal feeders is they attack the whole grains, because once the larvae, the adult lay the eggs on the grains, the larvae feed and develop inside the kernel. The primary insects are like rice weevil, moths, and as well lesser grain borer. Whereas the secondary pests, or they are the external feeders, that is red floor beetle, tribolium, you say. This is the one at the bottom of the thing. I'm sorry, I'm not able to use this, my pointer. The secondary pest is already, they feed on the already damaged grains by the primary pest. So this is the type of loss we get due to the insect damage. So the losses, usually they are divided into direct losses, and we can say the indirect losses. Direct losses is because the insects feed on the grains, there will be a reduction in the weight of the grain, loss of nutrients, and sometimes if you're going for a seed purpose, the germination is getting affected, and the grade is also reduced. So overall, the lower market price because of this insect contamination. Indirect losses is due to insect contamination. You will also develop the fungal problem, and you start going for more control and application costs, excessive pesticide residues, and these are the types of losses. What are the main causes for these things? Is first thing is moisture content. I think many of the speakers were would have been talked on. Moisture content is the major factor limiting the storage in case of millets. The next one is the temperature, the time period, how long you are storing it, the period of storage, the field infestation. These insects directly, they cannot come and damage into your storage bins or the bags, because they get infestation from the field itself. And sometimes in the cracks and crevices and the containers, bags, what does I mean here is the already used bags. Some grain is left, the new grain is also, the farmers will sometimes get mix the old and new grains, then there will be a problem. And the trucks and trolleys or the bullock carts during the storage, these are the important factors causing for the insect damage. So how do we go for the management of the insects? The first and foremost important thing is the sanitation or the hygiene conditions. The second one is frequently monitoring the pest situation, and the third one is the aeration is very important. Physical methods, I'll be telling what are the things. And finally, if it is not controlled with all these things, it is chemicals or the last resort. And I insist here that chemicals we don't directly apply into to the grains and all that. We can go for go down spray and all that. I'll be telling on that. So aeration is proper aeration of grain. Usually it ensures if you aerate them uniform temperature and avoids the moisture buildup so that the mold development will be reduced. The second point is the grain moisture. Always the moisture should be less than 12% or 10%. This is the moisture meter. You can measure with that. As I said, the storage hygiene conditions, wherever the millets are stored, it should be clean. Clean storage rooms. And the point where I have highlighted is don't mix the old grain and new grain together, because the old grain, if in case it is already damaged by the insects, there will be an easy spread of to the new grain also. So how do you monitor these insects? Here I'm showing the two methods, like sampling and the probe traps. The sampling is there is a sample drawer. You can just put it in the probe should be inserted horizontally 5 centimeters under the grain surface in the center of the bin where the grain surface has not been disturbed. So the other one, then the one which is the probe trap, here you have to insert it vertically with the plastic, this white plastic cone inside, downside the grain. So that red top cap must be level. This red top should be with the level of the grain. So insects, what they will, if at all they are there, they will move towards air in the main tube and enter through the hole. So once the insect enters the hole, it falls into the detachable white cone, and this white cone can be unscrewed once in a week and you can destroy the insects. This type of probe traps, you can insert 2 to 3 numbers per 25 kgs of the bin. And the next one is disinfestation of the storage. What I was telling is this is the infestation of the go downs. You can apply a safer insecticide like Malathion 50 EC, 10 ml per liter of water, and 3 liters of spray solution is required for 100 square meters. This, I'm insisting that it should be only disinfestation of the go downs, not with the grains. Sometimes the activated grain protectants are also there, like activated clay or diatomaceous earth. This dust will absorb the epicuticular waxes of the insects, causing dehydration, and there will be death of the insect. That is the mode of the action. So even gunny bags, I said if some problem is there, the old bags, you can impregnate with 0.1% malathion for 10 minutes, and you have to shade dry them to give the protection to the shade. After the shade drying only, you can use those bags. The final thing to control the insects is like fumigation. In market, there are available sulfose tablets. In the aluminum phosphide is available in sulfose tablets. There are two methods. You can go for shed fumigation or the cover fumigation. Shed fumigation is nothing but you can fumigate the entire go down. Here you can use 21 tablets. Each tablet contains 3 grams of this aluminum phosphide. It is sufficient for 28 cubic meters of the storage go down, and you can fumigate for 5 days. You insert the required number of aluminum phosphide tablets in between the bags. So cover the bags immediately with the fumigation cover. You can see this. This is the sort of fumigation you can do. So now, is it possible to control with one method or the 2-3 methods? Here at IIMR, we are trying to study in an integrated approach. Like if you want to protect a grain or the floor, first thing to control is the moisture in the grain. So we are trying to do the moisture and the drying techniques. Then we are going for to control the insect exert. We are going for the radiation, gamma radiation, and other processing in techniques, and finally the packaging. All this together, integrated package, we can enhance the shelf life of the millets, grains, as well as the flour. Now, when we talk about the shelf life, there are 3 things here. Shelf life, we talk about the shelf life of the grain, shelf life of the flour, and the shelf life of the products. So shelf life of the grain, how to can be increased by through management is through, I was talking about optimizing the moisture content, irradiation, that is gamma radiation, and packaging is also a very important technique to prevent the damage by the insects. And the processing, here the management, the processing techniques we usually apply is the moist and dry heat techniques. The next one is floor. Floor, when we talk about the rancidity comes into the pictures. What is rancidity? I will tell you, this is because of the floor develop the off flavor due to the volatile organic compounds or the enzymatic degradation and the lipid composition of the grains. And so this floor increase the shelf life can be done with the packaging materials like modified atmosphere packaging or the vacuum packaging. I'll be dealing in the other slides of them. So what are the interventions we are doing for controlling the insect damages? First thing, optimizing the moisture content, packaging, then radiation, as I said. So how do we optimize the moisture content? Usually we suggest for sun drying if there is a big lot of samples. For a smaller samples, we can go for existing moisture content. You have to check what is the existing moisture content, and you have to condition your grains to less than 12% or 10%. So either you can do it by sun drying or using by the ovens. If it is a smaller samples, you can go with oven. If big larger samples bags, you can put it in the sun and then check with your moisture meter. They are very cheap. So we are at IEMR, what we have done is we have taken 3 levels of moisture contents. Uh, I'm sorry that I don't know why this slide is missing. You can see we had tested the existing moisture content and the conditioned moisture content. If you see the existing moisture content, the insects have developed, but in the whatever the optimized moisture content of 12% and 10%, you can see there are no peaks here. There is no insect development. So pearl millet can be stored more than 18 months without insect infestation at optimized moisture content of 10 to 12%. Similarly, foxtail millet also we have done. Here only in the existing moisture content, we have the insect damage. And even the ragi, finger millet, we have tested here. And the sorghum grains also. So I am saying here 18 months, it can be more also because my experiment was, we checked it for 2 years. So the optimized moisture content, when you do it below 12% or below 10%, there is no insect damage up to more than 2 years. The other technique what we have used is gamma radiation. Here about 1 kg of millet grains, we have packed it in a polythene bags and we have passed through the gamma radiation. And the doses we have tested is 200, 400, 600, 800 and 1000. That is lower and higher doses. And unirradiated seeds served as a control. So each genotype was replicated thrice. Here also we have tested for all the sorghum, pearl millet, and small millets, finger millet and foxtail millet. Then we have taken the data on grain damage, weevil development, and all that. See, in the graph, we have depicted the different millets, pearl millet, foxtail, finger, and sorghum. When we have irradiated the grains of this, you can see that like in pearl millet, the grain weight loss and all that, at lower doses, we have 200 was not effective. These are the control ones. And in 400, it is safe. So any of these millets, when treated with gamma radiation of 400 GY, can be stored for more than 15 to 18 months without insect infestation. So the other third method, first one I have talked about the moisture content, then the irradiation. Gamma radiation, why we go is, it kills the, if any eggs are laid on the grains or the larva is there, the gamma radiation will kill both the eggs as well as the larvae. So we go for that gamma radiation. And the third method is packaging is these days very important. In gunny bags, you get frequent insect infestation. We have tried different types of packaging like LDPE, HDPE and laminated pouches and control pouches. Laminated is you get 3 layers. Here LDPE, you have this only 1 layer of LDPE polythene. Here in laminated, you have 3 layers. So this what we found is all the three packaging is very good for insect infestation. But in case of floor, this laminated pouches were more effective when compared to the other packaging. The other method is modified atmosphere. This is nothing but into your packaging material, you allow the nitrogen gas into that, so that you insert the inert gas, that is nitrogen, and this all millet genotypes we have tested into all this packaging material and found that this is very effective when compared to the normal polythene packaging. Another one is vacuum packaging, and the similar way we got a very good results here. So here I'm comparing the control packaging, that is like your normal polythene, and our HDPE or any other packaging. No insects were observed even after 10 months or more than 2 years. This is all about the shelf life, how you can enhance the shelf life of the grain. But now coming into the shelf life of millet flour. If you see, I just put the structure of the millet grains. This is the pearl millet, next this sorghum. It is called, both of them are called as caryopsis. It is covered with pericarp, germ, and endosperm. Why this I will tell you, these are important in when concerned to the shelf life of pearl millet. And the other millets, minor millets, foxtail, and the other right side is the finger millet. The pericarp in finger millet, you have a grain pericarp consists of 3 layers. So very strong layers. So the damage caused in ragi, finger millet, by either by by insects or the enzymatic degradation is very less because of this hard structure of the grain. So what makes the poor shelf life of the pearl millet? If I say that pearl millet is very rich in lipids, they are concentrated in this germ. That's why I've shown the structure of the grain. The germ, you have the three parts, no, pericarp, germ, and the endosperm. So the lipids is nothing but fats or concentrated in the germ. And all the enzymes responsible, whatever the enzymes responsible for hydrolysis and oxidation, are in the endosperm. So while making flour, as long it is a grain, it is safe. Once you mill it, once it is made into a flour, they get mixed, and lipase, the enzyme lipase, act on the lipids to produce fatty acids, which are oxidized by other enzymes like lipoxygenases and peroxidases, so to aldehydes and ketones, having a foul off flavor smell. So this is called the rancidity. Oxidation also takes place both enzymatically as well as non-enzymatically. Non-enzymatically is because of sunlight and oxygen. So rancidity is a natural process of decomposition of fats and oils by hydrolysis or oxidation or both. So why I'm insisting on this is, like I said, millets like sorghum and pearl millet have high lipid material, and almost pearl 1 to 5%. Pearl millet is having the more lipid content, and the germ comprises of high triglycerides and all this. These are very important to study on the rancidity. So FFA is nothing but free fatty acids. I said during the rancidity process, the free fatty acids are released. So free fatty acid content is a basic indicator of rancidity. How do we measure rancidity? How do you say that flour is become rancid or less shelf life? One method is organoleptic, just taste it and tell. But scientifically, you can prove that what are the changes are happening here. So in that way, as I said, pearl millet or any other millet, rancidity is of two types. This is oxidative rancidity and enzymatic rancidity. So because the pearl millet is rich in lipids, that is fats, once it is milled to flour, it starts now the enzymatic rancidity. So the free fatty acids are released. In the presence of oxygen and light, you have this oxidative rancidity. In the presence of enzymes like lipase and peroxidase and polyphenol oxidase, you get the enzymatic rancidity. So final thing is off flavor, causing compounds, free fatty acids, bitter taste, all this. So pearl millet cannot, flour cannot be stored not more than a week. Sometimes people say it gets rancid even at 5 days also. So my point here is controlling one is not sufficient, either oxidation or enzymatic. So you have to control both the oxidative rancidity and enzymatic rancidity. So here, yeah, this is an repetition. Yeah, so in that process, we have also studied what is the lipid content and composition in the millets of this pearl millet, foxtail, finger, and sorghum. If you see the fatty acids, the major quantity is up to 5 in case of pearl millet. Next, for in foxtail millet, it is 3 to 4, finger millet 0.7 to 1.5, and sorghum this much. And if you see the composition, like saturated, unsaturated, and PUFA. PUFA is polyunsaturated fatty acids. Wherever the polyunsaturated fatty acids are more, it is prone to um, the grain prone to rancid very fast. If you see, but you can ask me a question here. In finger millet, it is less. In foxtail millet, it is more than the pearl millet, but pearl millet is getting spoiled faster than foxtail millet, even though the PUFA content is more in foxtail millet. Yes, we have a reason here. If you see, the PUFA content is more in pearl millet, as well as the lipase activity is also very high in pearl millet. In foxtail millet, PUFA content is more, but the lipase activity is medium. In case of finger millet, both PUFA as well as the lipase activity are less. That's why the finger millet grains are stored, or the flour is stored for longer duration when compared to the pearl millet and foxtail millet. So these two are more important, the fatty acids as well as the lipase quantity. So now we know the cause, we know how the flour is becoming rancid, what is the reasons. So now coming to the treatment methods, how to enhance them. There are two methods, traditional and conventional methods, and the other one is advanced or the novel technologies. When we come to the traditional methods, we have different types of processing technologies like mechanical processing, simple fermentation, germination, thermal treatment, and addition of preservatives. So even in thermal treatment, again, we have two types, the dry heat method and hydrothermal treatment. Now coming to the advanced technologies, we have thermal and non-thermal treatments. In thermal, we have microwave processing, infrared processing. In non-thermal, we have gamma radiation and high pressure processing. These are the technologies. Now coming to the individual thing, I will just give a brief introduction. Like the traditional processing, like mechanical processing, we have malting, fermentation, as I said, soaking. So first method is decantation, is nothing but removing the outer part, that is the husk, so when you remove the outer husk, it increases the storage stability of millets. Why? Because decantation considerably, it will decrease the fat content of the grain. So this is the preliminary traditional method what we were using now also. So the next one is the fermentation method. Fermentation, different methods. You have using the yeast and lactic acid bacteria. It's have a symbiotic relationship. So that methods fermentation also will increase the shelf life of the millets. The other one is malting, like the steps like soaking, then controlling the germination, and then drying. These traditional methods will increase the shelf life of the millets. Other one is using the acid treatments like antimicrobial, antioxidants, acid treatment with 0.2 NHCl. So that when we use these things, we have a reduction of 70 up to 74% of reduction in free fatty acids. The other one is antioxidants can be used. So many antioxidants like BHT treated flour, but the lower quantity we have to use. Even when a test, an experiment indicated that 0.02% of BHT treated flour of pearl millet increased, the FFA was decreased here. So we have naturally products like derived from plants like essential oils, from plants like rosemary, lemongrass, citronella, cinnamon, basil. These all formulations, these days they are gaining importance because of natural things, and these can be used as preservatives also. The next, conventional thermal methods. This is very important. Dry heat treatment and hydrothermal treatment. Hydrothermal is soaking and then drying. Dry heat is only heat treatment. Here dry heat, you can go for solar drying, or you can go for oven drying. This will inactivate the spoilage of the microorganisms. For example, in sorghum, it was when tested for foxtail millet, pearl millet. When we have done for dry heat treatment, significantly it reduced the fungal counts also. And what will happen? There are two-step methods in reducing the FFA content when you go for dry heat treatment. So what I said, rancidity, how do you measure? One is organoleptic tasting the thing. The other scientific thing says the parameters, or there are 4 parameters for rancidity things, how to test. One is alcoholic acidity. Yeah, this is nothing but it refers to the combined acidity. Hydrolysis, I said no, that is hydrolysis of fats by lipases into free fatty acids, one thing. So when we have tested pearl millet, if you see, I have given example of pearl millet. When pearl millet is stored from up to 60 days, the alcoholic acidity values are increased during storage continuously. From 0 to 60 days, it continuously increased. And then down graph is on different genotypes. So the variation is there among the genotypes also. The second one is the rancidity parameter is free fatty acids. So how do we measure them? The release of free fatty acids. There are different procedures here. If you see the free fatty acids also, it increased during the storage from 0 to 60 days. If you see, the values are increasing. And here also among definitely there are changes in the genotypes. The third one is the peroxide value. The similar thing, it is nothing but peroxidation is deterioration here, reduce the stability and formation of, these are majorly of flavors. So peroxide values are also increasing during the storage from 0 day to 2 months. We have studied up to 2 months. So the genotypic differences are also there. The final enzymatic, also lipases also, if you see, it is increasing during the storage. This is about the rancidity parameters. Now, what we have done is, as we have done an integrated approach for the grain, increasing the shelf life of grain, we have done it for the shelf life increasing for the flour also with integrated method. The integrated method here is we have compared, the first column is the control with other laminate packaging. We have taken the laminated package along with thermal treatment, then lamination with the modified atmosphere. We have used here nitrogen, it is an inert gas. Then the third combination is the packaging, thermal treatment, and the modified atmospheric package. If you see the values, when in 0 days and 7 days, whatever the value of alkalic acidity is up to 7 days, 0.3, we could bring down that value with our integrated packaging, 0.33. If you see up to 60 days, it has gone, the value has gone to 1, where we have brought it with our integrated package to 0.3, that is 7 days interval. Similar cases with pearl millet, foxtail, finger, sorghum, and we have done for several genotypes. And we have concluded that the integrated package of lamination, thermal treatment, and the modified atmosphere will reduce the alcoholic acidity as well as free fatty acids also. If you can see, initially it is 70, up to 60 days, it has gone to more than 2000. These are the values I've given. The how do you measure it? And our integrated package, if you see, it is 151 even after storage for 60 days. That is, we are bringing less than 7 days. So we are increasing the shelf life and showing it in through biochemical matter, parameters as well as the organoleptic properties also. So for alcoholic acidity, we could reduce it. Free fatty acids, we could reduce it. Similar way, the peroxide values also. If you see after 60 days also, our integrated treatment with lamination, thermal and nitrogen could bring the values to 5.1. That is whatever it was initially at before 1 week. So this was, we have done it here also for all the millets. And we have done for lipases also. The similar one, we could bring it up to the 1 week thing after the 60 days also. Finally, what we, after our study, we confidently could say that integrated approach of this is thermal treatment and storage in laminated pouches using nitrogen atmosphere, it has decreased the free fatty acids value by 10 to 15 fold, peroxide value by 2-fold, and alcoholic acidity by 2 to 3-fold, and extended the shelf life of pearl millet, foxtail, and all sorghum also for more than 2 months. The other methods I will just go through. I think I'm taking longer time. The other hydrothermal treatment, where we go for blanching, steaming, and boiling. Now coming to this or the normal, now coming to novel thermal processing techniques. I will tell you, these are the techniques recently introduced, but there are a lot of research is still needed. It is only to say that what are the advanced technologies are going to be available. It has still in under the research only. It has not come into the commercialization. One is microwave processing. Second one is infrared processing. Third one is heating and radio frequency heating. These are all still under research. So the difference between the traditional and the advanced technologies is the time taking there is more. Here it will be done microwaves when we are using it. You can do it very fast. Even the infrared rays, this can cause effective inactivation of the lipases will be faster here, and the free fatty acids also. So what are the advantages of these new technologies is they possess a fast heating rate. They could significantly reduce the consumption, the energy consumption, the techniques of high thermal efficiencies, there will be uniform rise in temperature, better retention of nutrients and all those things. But the disadvantages are also more here. The high initial capital investment, as I said, lack of in-depth understanding and experience still under research, but it may come fast. Then the other methods are gamma radiation, high pressure processing, pulsed light processing, and ultrasound processing. I'll just go through. These are the technologies available, recent novel technologies like high pressure. All the technologies is inactivation of the lipases and reduction of the free fatty acids. For this only these technologies are used. If you see this non-thermal processing technologies here, another one is ultrasound processing. Yeah. Now we have dealt about grain, flour. Now coming to the products. Here the pulp millet products means you don't directly treat the products. You will treat the grain only. Even for flour also, we are treating the grain and making the flour. The pulp millet grains, when subjected to two treatments like blanching, roasting, and drying here, the blanched pearl millet flour could be stored for more than 75 days in refrigerated conditions. When we use the blanched and roasted pearl millet flours, stored in refrigerated conditions, I said no organoleptic properties. It could be stored for more than 3 months. So finally, what I will conclude is, if you see the millet grain flour value-added products, the existing and after our intervention, we have increased the grain to almost 2 years, flour to 2 to 6 months, 2 months, value-added products to 3 months, and sometimes the snacks, like extruded snacks, we have increased to almost 1 year. So we are trying to increase the floor's self stability also for more than 6 months with our interventions. Thank you.